Sir?
Vidya ma'am will start the meeting.
Vidya ma'am.
Yes. Okay.
Okay, sir.
அவங்க பக்கத்துல தான் இருக்காங்க. Question answers நீ பாத்துக்கவேல?
Sir, yes, sir.
Okay.
மச்சான் என்ன நீங்க?
Vasant?
Sir, yes, sir.
Please check the live streaming.
Yes, sir. It's going on. Hello? Arunana?
Yes, Guru. Guru, I am here.
Yeah. Okay.
I asked Vasant to check. Yes.
முத்தமிழ் வந்தாப்லீங்களா?
இருக்காரா?
Yes, I'm here.
Yeah. முத்தமிழ் சார் வணக்கம்.
வணக்கம், எப்படி இருக்கீங்க?
நல்லா இருக்கேன், நீங்க எப்படி இருக்கீங்க?
நல்லா இருக்கேன்.
Right. Okay, fine. Very happy, sir. Happy to invite you in this webinar.
I'm so happy. I'm too.
Thank you. Take part in this session.
Definitely. We can.
Yeah, drink பண்ணிட்டீங்க. We'll start.
Yes.
Allow பண்ணிடலாம். அண்ணா அருண் சார் காணுமே.
அது ஸ்டார்ட் பண்ணுங்க.
அவருக்கு ஓ போஸ்ட் கொடுத்தீங்களா?
கொடுத்துட்டேன். குருசரவணன் அங்க இருக்கேன்.
ஓகேண்ணா. லைவ் ஸ்ட்ரீம் வருது இல்ல? லைவ் ஸ்ட்ரீம் யூடியூப்ல. எல்லாமே கரெக்டா இருக்குல்ல? பேசுறேன். அருண் சார் Tamil sir, shall we wait for 5 minutes? The participants are coming.
No problem. We can wait for. I have no work.
5 minutes. Okay, sir. Okay.
I think they are not able to hear you.
குரு கேக்குதா? கேக்குதா? ஹலோ முத்தமிழ் கேக்குதா?
கேக்குதுண்ணா.
எஸ். பிகாஸ் சம்டைம்ஸ் திடீர்னு ஆடியோ மியூட் ஆயிடுது.
அப்போ போய் செக் பண்ணீங்க.
எஸ் எஸ். என்ன தெரியல குரு டக்குன்னு ஆடியோ மியூட்.
Arun sir?
Yes, yes, I'm here.
Now shall we start the program? Start the session. Yeah, madam.
Good evening, everyone. Welcome to the next interesting and exciting session. And for this, I request Dr. Arun Muthukrishnan, Assistant Professor, Department of Biotechnology, Bharatiya University, to introduce the speaker. Please, sir.
Can you hear me?
Yeah.
So now I'm very happy and I'm really proud to introduce my brother, Dr. So I should say, PhD in
Your voice not clear. Arun sir?
Can you now hear me clearly?
Not audible.
Can you now hear me clearly?
Okay, okay. Yeah. Go ahead.
Okay, sorry. Shall I speak? Is it clear, Dr. Guru?
Yeah.
So,
Okay, now. Okay.
I'm very happy to introduce my brother, Dr. Selva. So he did his PhD in biotechnology in National University of Taiwan. So his
Arun sir?
PhD, yes.
Arun sir, sorry. Arun sir, please check your mic, sir.
My mic is okay. Now?
Now okay, sir. Continue with this.
One second. Guys, please switch on the fan. Sorry. Okay, now it's clear?
Yeah.
Clear.
Okay. So like he did his PhD in National University in Taiwan. In his PhD work, he developed transgenic cells to produce chimeric bamboo like particles. It's like for like an animal vaccine, I think. Yeah. After completion of his PhD, then he joined in the reputed institute in Taiwan, like Academia Sinica in Taipei, Taiwan. Like he continued his postdoctoral fellowship there, and in the Department of Herbal Medicine, almost 3 years, Dr. Muthamal is working there as a postdoctoral fellow, and actually there he is researching trial of medicinal plant. Amylosis in boilers. So that was his first postdoctoral. After his first, next he moved to. Then I think it was in South Korea. I think. Supervision of Inwa Hong. The second postdoc was like from there. In the second postdoc, plant-based vaccines for
Arun sir,
yes.
not audible, not clear your
I'm
voice.
trying my best. I'm trying my best.
Please.
Please. Okay. Hello.
Yeah.
Is in continuing under the same. So that is now in the development of plant-derived biopharma. So that is was now he is currently working. So he's a very eminent scientist. He is working in the plant molecular farming for a very long time. So now I request Dr. Muthan to continue his. Yes, Tamal, please.
Okay. Thank you, doctor. Thank you very much for your kind introduction about me. Good evening, everyone. So, dear professor and research scholar and students, thanks for having your presence in the wonderful evening through online webinar. So, I also thank to Dr. Arun and Dr. Gurusaravanan to organizing such a wonderful international conference through online during the pandemic. So today I'm going to discuss about recent trends in plant biotechnology. In plant biotechnology, so far it has the evolution from the plant cell culture, which used for the plant secondary metabolites production. From that onwards, it gone different kind of evolution to reach today level. So why is plant molecular forming so very important? What is the use of plant molecular forming? So that is the most important question which we need to answer that. In general, we use plant as a bioreactor. So before every 10 years, there is a new technology coming up and the old technology will be gone. Otherwise, every 10 years coming new technology to alternative for the conventional based technology which used for the production of biologically important proteins, enzymes, or vaccines. This is a trending formula. But recently, most of the biotechnology researchers, they are focusing on use the plant as a bioreactor to produce various kind of biologically important proteins, not only for the research but also they want to make that, or they make as a product for the industrial pharmaceutical industries. So what is the main objective of this plant made pharmaceuticals or plant molecular forming? Is it really important, or is it really replace our existing eukaryotic production system? For instance, bacteria, yeast cells, and mammalian culture. These are used for the production of various kind of proteins, enzymes, or interleukins. So there are a lot of things they use as a bioreactor to produce or to for the industrial purpose or any medicinal purpose or vaccine, whatever. So they're using for that kind of purpose. So why most of the researchers focusing on plant? It's a very simple answer, because most of the Asian countries or third world countries, they are most of the people are very poor. They cannot afford the drugs or pharmaceutically important proteins produced by mammalian cells or bacteria or yeast yeast system, because those are very, very expensive. The plant, you can have a small greenhouse, you can produce with the high biomass, because you can have the tons of biomass. We can easily purify the our protein of interest. That's the only reason, because plant can make very simple, very cheap, and a low cost for the production of any biologically important proteins. Okay, so what is the molecular forming? That's a very simple answer. Is bioforming is a very recent trend in biotechnology. They use genetically modified plants which can produce recombinant proteins or pharmaceuticals, even industrial enzymes. Not only that, also we can produce vaccines using the plant as a bioreactor. So you can see here a simple aseptic condition. We can grow the plants, which be a transgenic or transient. So I will explain to what are the different kind of systems available to express this kind of enzymes, pharmaceuticals, and recombinant proteins. So very simple is use as a plant, genetically modify. From that, we produce the protein inside within the cell, the break the cell and purify using different kind of columns or tag or whatever we can have the available different kind of things. We can purify it. After purification, make it as a products to commercialize for the beneficial of human beings. That's it. Very simple. So why? What is the advantage of plant-based expression system? Why not the yeast? What is the different? Uh, what is the most important factors which is involved in the plant system in compared with yeast, bacteria, or human mammalian cell systems, or any other eukaryotic production system? Because this plant-based system can be amenable to the current manufacturing practice CCMP. They called CCMP. So any proteins or any drugs which it, developed by country, they should follow a set of regulations, which very important to uh, any side effects, that drug having any side effects or it has any causative agents, that kind of things we need to be very clear about that. Then only they will allow to you to use in the public forum or commercially. Second important point is we can produce a large amount of biomass. For instance, just 20,000, just 1 acre land, like that. You think about 1 acre land, we can produce large amount of ton amount of biomass that can be harvested, can be easily break a purified protein from those using the biomass. Second important point is very easy to express wide range of proteins, wide range of vaccines, or any industrial enzymes. So nowadays, industrial enzyme mostly they expressed using the bacterial system or yeast system, which costs a lot of money because we need to use a big bioreactors. Whereas in industrial enzymes, which are expressed in the plant system, it's a very easy, very simple. So just to put your gene of interest into the plant cells, it can express just to crush the you no need to purify, even you don't need to purify because industrial enzyme no need to very highly purified form, just to break the cell walls and extract the protein, and directly you can use for the any industrial purpose. For instance, biomass waste management, or any textile industry, or any paper industry, you can directly use after the expression in the plants and directly extract the soup from the plant cells. So very simple. Another most important point is the whatever enzymes. For instance, if you express any proteins in the eukaryotic bacterial system, most of the proteins, mostly even most of the human protein mostly will not have in the glycosylation. So that's the most important point, because without the glycosylation your protein cannot have the proper structure. So whereas in plant system, it's like a mammalian system, eukaryotic system. So it can retain native protein structure, but a little small modification, with a small modification. But still 99% is the you can get the right pattern of the right structural protein, or with a very active form of protein you can be purified. Also, you can eat the plants directly through yes. You have any question? Okay. Can you hear me right?
Yeah, good.
Okay.
Go
Okay.
ahead.
Okay. Okay. So plant-based system, there is no determinable cause. Like when you use the yeast or bacterial system or mammalian system, basically when you purify the any drug or protein, it can also purified with some toxins or viral toxins. It causes some side effect to the recipients. Whereas when you express any protein from the plant system, it will not cause any kind of detrimental or any kind of side effect, because we just eat the plants, as like we eat a lot of tomatoes or we eat a lot of fruits. We'll eat even neem tree, we just eat, right? So it will not cause any kind of side effects for the humans. So when you express this kind of plant as a bioreactor, using that we can express any kind of protein that will not have any impact on human health. So that is the most important point of why we use plant as a system or bioreactor to produce any kind of biologically important proteins or vaccines or industrial enzyme. And that is a major advantages over any other eukaryotic system which have been used for the producing those kind of things. Okay, now I just generally talk about the what is the advantage of plant system. Now we need to know how. Okay, we told about the plant system, right? But how do you express the protein in the plant system? That's a most intriguing point. Okay, what are the technologies available? What kind of strategy? Because one strategy always not give answer to the all the problems or all the things. So we need different kind of strategy to produce large amount of protein in plants. So that is the most important goal in our study or research. Okay, here you can see here there are two major division in the expression system in the plant. What are the two major expression system? One is transient expression system. Another one is called stable expression system. You know that what is the meaning of transient or stable? The stable means your gene of interest, which integrated to the plant genome, which develop as a transgenic plant, which you can every time you collect the seeds, you can show the seeds and collect the leaf, and you can produce the protein. It's a stable, consistently it produce the protein as long as that gene integrated into the nuclear genome. In that stable expression system, there is another called transplastomics. We called it transplastomics or chloroplast transformation. What we'll do, the same gene of interest, instead of nuclear genome, what we do, the gene of interest integrate into the chloroplast genome, right? Because we have a lot of chloroplast in the single cells, so you can get more and more protein you can get. So that's why we also divert to the chloroplast. So only one problem in chloroplast, some of the protein may not have the glycosylation, but most of the we don't need all the protein need to be glycosylated. When you need the large amount of protein, you can also go through chloroplast transformation to express your gene of interest in the chloroplast, or you can also transfer your gene of interest to the nuclear uh, genome and plant's nuclear, so that it can express. So in the nuclear genome, you can also target any places. Like if you have the marker which you need to express only leaves, okay, you can put the leaves. Or you can want to only store your proteins in the states, okay, you can also target the by using the marker, you can target to the seeds. When you mature the seeds, most of the protein will be stored in the seeds. When you want to do the in the edible vaccine or edible protein, you can also target it to the fruits. It depends on as to decide how what kind of things we need, what kind of things also we need. So another important expression system called transient expression system. Transient expression system is based on the plant virus system. Also, we can use the binary vector, Agrobacterium tumefaciens, right? Which we used as a carrier to transform the stable transgenic plant, or this transient expression a very short while. The tDNA will go to the nuclear. From that, it will transcribe and send to the cytoplasm, and it will do the jobs. So plant virus, as you know, I don't know how many of you very aware about the plant virus and the plant virus based expression system. So the plant virus expression system will produce a large amount of protein within a very short period of time. The difference between stable and the transient expression system: stable expression system, you need to spend 2 years, 3 years to develop the transgenic plant. So for instance, any pandemic, we cannot rely on the stable expression system. Whereas, for instance, now we have the COVID-19, we need to produce some kind of protein, some code protein or some kind of S protein, S1, S2 in the COVID. We need immediate because it's a pandemic situation. So for that kind of situation, transient expression system, the best system. Just when you design the expression cassette, put your gene of interest and express immediately within 3 days, 5 days, based on the accumulation of the gene of interest, you can produce it in use the plant-based system. So in plant-based system, there are two kind of expression system. I will talk about. We will discuss about that later on. Okay. Apart from that, also we can use the root system, hairy roots. Especially Dr. Arun is an expert in this hairy root culture. So we can target our gene of interest into the hairy root culture. So we can also express in that. But recently we only transit expression system, stable expression system only virus in leaf or chloroplast. This is most of the predominant researchers focusing on this two part because it can very easy and very convenient. We don't need any expensive material to produce any pharmaceutical proteins. Okay, that what I said it. Advantage of what is advantage of stable expression system or agrobacterium based expression system or transient expression system. Disadvantage of stable expression system is the production rate is less than 1% or maybe right now until 5% is reached the expression system. But disadvantage of that, but advantage is consistently it can produce. It can scale up to how much amount of biomass you can produce. You can definitely produce. But advantage of transplastomic transformation, or it's called chloroplast transformation or chloroplast transgenic, it can produce 34% TSP, means total soluble protein. When you having the after extraction, you have the total soluble protein, right? In that you can say 34% until 34%. You can get your target protein, any protein, like any kind of protein you want. For instance, S1 protein, S2 protein from the COVID. But we didn't test that one. But I'm saying so far they reached 34% about a protein of interest. Agrobest is rapid, but the protein, sometimes the accumulation of foreign proteins or pharmaceutical proteins are very, very low. It's not very high. Plant virus always provide a very better, best, high accumulation. Sometimes protein to protein, it may vary the expression level, but still the plant-based expression systems are the best expression system. Most of the people are using for the even if you heard about the Ebola vaccine, very rapidly they produce the Ebola vaccine, right? That using the plant virus as a system to produce the Ebola vaccine within 6 months they produce it. So that's how they produce. Okay, so now it's how very brief. Before I just said about how the briefly about a stable nuclear system or transplastomic genetic transformation. So here the how they are doing the transplastomics on the stable nuclear things. So this is a bacteria. It's agrobacterium tuberculosis. Okay, what they do just to they transform the binary vector. I know you all know it, the binary vector. So they put the TD in between the tDNA, your left border, right border. They put your gene of interest. Then they transfer to the using the agrobacterium. They transfer the nuclear through the gene of interest to the nuclear. So once the nuclear transfer, they have a series of selection and regeneration. After regeneration, they grow in the acclimatization in the greenhouse. After greenhouse, so if you are your protein expressed to leaf, they make it a powder or whatever. If you want a purified form, you can take the purified form that go to the HPLC or any columns like affinity columns or any kind of columns. Maybe you know that nickel NTD column, his tag or a MIG tag, any tag you can put the your protein of interest. You can use that one. You can easily produce the protein that can be mass level we can produce. It's not a if You just think about the if you want to grow the bacteria, you need a 1,000 liter tank. For that, you need to controller. You need to see the cells should be multiplied. So those lot of procedure available need for the bacterial or animal cells or yeast cells. Whereas plant, it doesn't matter. Just you need 25. You have to maintain the humidity and the temperature in the greenhouse. That's the only important. So if any infection there, just immediately remove that plants. So remaining plants will be saved. So that is the advantage of plant-based system compared with the yeast system, or bacterial yeast system, bacteria, or any eukaryotic system. So that is the advantage. So it can be used for any kind of things, any kind of protein, neurology or gastric problem, or any kind of things, you can easily go for the plant-based system. So here the two system, how they transform the cells, and from that how to go the refilling the drugs, and the drugs from that how to go for the commercialization of this kind of things. Second is the of course I earlier said the same way, transient expression system. How they do, you have to know how to do the transient expression system. This is called agro infiltration. In this syringe having the agrobacterium soup. So they mixed it with some kind of mist and some kind of chemicals. Then they make it the also add the acetylcholine as a chemoreceptor for the transformation of your gene of interest. Then they use just a small syringe, and backside they will press the agrobacterium, transform spread across the cells, so it can be easily produced. So once we harvest the cells and just non-filtrated, infiltrated, just extract the proteins. From that, clarified and use any kind of columns. Any kind of columns, you can use the affinity columns, or you can have some gel filtration. You can use the gel filtration. If your proteins are very complex, you can use affinity column, and you can use the gel filtration, again affinity, cationic or anionic, anything you can use for the purification. It's up to what kind of protein you choose, what kind of protein you want to express in the plants using the stable expression system or transient expression system. It's up to as to how to decide what kind of things we need to decide. So here I having the how they in industrial scale, how they produce the protein using the, so because there are a lot of companies today involving in the production of biologically important proteins. The companies, this is a manufacturing videos. There is a 4 minutes video. Maybe if you see this video, maybe very interesting for you to come to this field, because in future, though comparing with any eukaryotic production system, only plant going to be a future. So if you're maybe this video for you may be interesting, so I'm just playing this video. 4 minutes just to watch.
Developing the vaccine in this facility start from identifying the genetic material that is in bacteria or viruses. We take that genetic information, we convert it into proteins that we can express in our system, which is based on plants. The Fraunhofer Center for Molecular Biotechnology is the only not-for-profit plant-based translational research and development organization providing services for fast, safe, and effective protein production to support the development of vaccines and therapeutics. The center has more than a decade of success in collaborating with both public and private sectors. We provide a complete suite of R&D capabilities necessary to help clients navigate product development from proof of concept to phase 2 clinical trials, including good manufacturing practices, regulatory compliance, and technology transfer. Plants are some of the largest, fastest, and cheapest biomass generators. We demonstrated that our technology can make that process from unknown gene to known quantities of pure protein in tube in less than 1 month.
Each individual plant is literally going to express whatever DNA sequence we put in there.
We actually are using the plants as almost individual factories in themselves to actually make protein. So we sort of hijack their systems to make protein.
As
plants that are grown in each of the flats are moved into an area where they're vacuum infiltrated with agrobacterium.
Plants are actually submerged in a bacterial solution in the presence of a vacuum. We actually use the vacuum to force the bacteria into the plant.
Agrobacterium is the bus that moves our genetic material from the lab bench into the plants themselves.
The vacuum then degasses the solution. It also pulls the air from the intercellular space of the plant. And then when the vacuum's broke, the agro and the suspension actually rushes into the plant leaf.
That genetic material has been put in the plants. The plant's own machinery for making proteins takes over, and it makes our targets of interest, our vaccines and therapeutics.
After about 5 to 7 days of the plant acting as a bioreactor, we harvest the tissue. The tissue is then homogenized.
When they're harvested, they're cut right at the crown, right at the top of the plant flats. They're collected in a vessel, has buffer in it, and then they're homogenized. That plant homogenate is then clarified of any of the large particles.
It's almost like a green smoothie. And then from that, we do further purification to end up with a clear, colorless liquid free of particulates.
It's an extremely safe technology in the sense that we use no animal products while processing any particular antigen. We're really looking to make vaccines, for pennies per dose in order to supply and meet global demand and really help the world population.
Remember that vaccines are administered to healthy individuals, and in majority case, it's really children. So we want to make sure that you can tell the parents that there are reasons why you should take vaccines, and there are reasons why you should take this particular vaccine. The safety, efficacy, and potency of our products will facilitate development of pharmaceuticals needed to address public health issues of the 21st century.
I hope you enjoyed this video. So this is a very simple processing from the back infiltration, from that, how they end up with the product manufacturing. So there's a very simple, any protein or vaccine, protein, enzyme, anything you can, using the same technology, same things you can do. Okay, next we are going to talk about the plant-based expressions vectors, which can be used as a very good platform. Also, it is proven that this plant-based expression system can produce large amount of proteins in compared with the agrobacterium, which use the binary vector based methods. So what you do, that this is a copy of mosaic CPMB virus, or is called tobacco mosaic virus. It's a spherical shaped, it's a rod shaped virus. What do we do? Most of the virus, these are virus particle. It's a more than coat proteins. It's a shield which there are a lot of coat protein come joined together, form virus particle, in which N-terminal part mostly outer layer of the matured virus particle. So here we have the two kind of system to produce the virus particle. Virus particle. One is we remove the N-terminal or any part of small portion of virus particle, which remain as a healthy, and it can replicate virus in the plant cells. We put the small epitope like 50 more than 50 amino acid epitope, which can insert the N-terminal part of coat protein. Okay, for instance, here you can see the polymerase. It's a complete RDRP. It's a RNA dependent RNA polymerase, we call it, which are required for the replication of virus. So here we can, this is a RNA2, which is a pi parity. There are different kind of varieties of RNA, so I'm not going to talk about that because it is not today's topic. But virus also can be a monoparatite or diparatite or triparatite. So anything. So there are different kind of systems available. Here, copy of 6 are piparatite virus. This virus code protein, they insert any gene of interest. Here, N-terminal part. That N-terminal part can be easily displayed on the surface of a viral particle. So this virus act as a adjuvants. So adjuvants is displayed on the surface, which can have the more number of, for instance, there are 500 of monomers to form the virus-like particle. Means a single particle can have the 500 epitope on the surface of the virus-like particle. Similarly, tobacco mosaic virus, it has a rod-shaped virus. This virus also, every monomer of the code protein, it can display the epitope. So it means that the virus particle as a nanoparticle, as you know that virus is a nanoparticle, is about the 500 nanogram to nanometer to less than that, 250 nanometers. There are different kind of virus particle there. So based on that, which virus you are choosing for the express your gene of interest or epitope of interest, you can express in the on the surface of the virus particle. So it can easily enhance your immunogenicity, so that your body system react immediately. It can produce the B cells, induce the B cell to produce the antibody against your epitope. So one of the best system, it's called the antigen presentation particle, which is called antigen presentation. There is another called deconstructive virus particle. That means we completely remove the code protein region. Instead of that, we replace with our gene of interest, complete full length protein. You can also produce a small peptide, which can mostly used for the animal virus. In my PhD, I did the animal virus. I removed the 37 that's an amino acid from the my bamboo mosaic virus. In that, I put the FMD, foot and mouth disease virus epitope, VP1 epitope. I put there so that it can express and display on the surface of virus particle, which can strongly induce the immunogenicity in the swine for the swine. I mean, pig, for that pig vaccine I developed in my PhD program. So using the same plant viral based vector. So advantage of this virus, you can see here different kind of virus. It's called a spherical virus, it's called a rod shaped virus, it's called a flexious rod shaped virus. So these are the virus. So what is the advantage of plant based virus expression? As I said before also, it's a very extremely high level expression in compare with the transgenic plant or any other plant. Only also it can easily the you can produce a very active form of proteins. So this can be also very short period, within a short period like 3 days, 4 days, even 5 days, enough to produce your protein of interest. When you, if you design the proper way your gene of expression cassette with the necessary component, you can produce within the 7 days, 5 days, it depends on, even 3 days you can get the protein of interest within 3 days. So 5 days for another 5 days for the purification process. Within 7 days you can have the your protein of interest to check the whether this protein is active or whether this protein can induce the any biological activities or biological things. Also this protein also not sometimes also because it's a very hyper expression or a very high expression, sometimes plants feel this very hyper activity so that it can die some kind of die. But the only problem is this plant based butter, it's a very biosafety. So most of the problem is the virus is spread very rapidly, even just by touching it can, it's called trafficking, we say plant virus trafficking. They say it's immediately one cell to another cell to another, one plant to another plant to another plant. So there is easy to contamination, those kind of things. Only that's the only disadvantage based on the plant virus based technology. Whereas one of the best technology which most of the companies were instant icon, there are different kind of international reputed companies, they are producing a lot of proteins or pharmaceutical based on the plant virus vector based expression system. Okay, now I'm want to compare this. So now I'm talking with a different kind of expression system, and from that how gene of interest transform to the each system. Like I talk about that. Now I want to compare in the different kind of system, scale up and the quality, overall cost, how purification cost, contamination, this kind of things I'm talking about, comparing the different system. So you can see here. You can see here first plant cells. It's a plant cell. If you know that a plant cell suspension culture, you know that, right? The From the callus, we induce the callus from the leaf. That callus grow in the liquid medium. That's called a plant cell suspension culture. Also, so far we talk about only whole plants because I'm not focusing on the plant cell suspension culture, but I'm only focusing on whole plants systems. That's why I didn't talk about. But the plant cells suspension culture one of the best way, but it's take a lot of time to culture. A lot of contamination also will takes place. Whereas in compare with the whole plant system, you can see the different kind of system. For instance, bacteria, yeast, insect cell, all mammalian cells, or whole plant cells. When you see the protection times, it's a very long because you need to make the transgenic plant. But in case of transient expression system, it's a very just within 2 weeks you can get the products because this paper only talking about the transgenic plant, not transient expression system. But in case of transient expression, also same kind of quality has, but the only problem is it has some kind of cross contamination when you use the plant based plant vector based plant virus vector based system. Whereas agrobacterium, when you use that one, it doesn't have any problem. So this is a different kind of thing. You can compare with the different parameters for the very low, short, very low, or medium, high, long, from the product quality to contamination, purification cost. Purification cost is very low in the plant cells in compare with the other things. So only very high in the whole plant system, only production time. So that is one of the very highest part. And you can also use large amount of scale. You can see here very high scale. You can scale up the any protein or biomass here because you can get a lot of biomass. Also, overall cost also not too high. Still little high because of still nowadays we have a lot of different kind of technology. This is 6 years before this paper came, so they didn't add this point, or I didn't put the recent review articles. But this recently we have very potential and a very great papers are available, research already out, and we can reduce the even longer time also. We can reduce even overall cost also. We can reduce in the using the plant system and compare with the other system. So cost wise, if you see the very low cost, very low compared with any other system because it doesn't need any sophisticated system or sophisticated bioreactors or anything. You don't need, just you need 20. You have to maintain the 25-degree in the greenhouse, so plus or minus one. It doesn't matter. But you have to maintain the right humidity. So humidity is also very important to grow the plants. So you also take care of plant health. That's also most important. Then you can get the right product, high quality products within the short period of time. So that's the most important part. Contamination, mostly contamination risk is a very less, but comparatively not the problem. Sometimes purification, yes, you need to use some. Right now we have the very simple purification tag like cellulose binding domain. We have small protein drug, but maybe I didn't add here because that's research oriented. I'm saying cellulose binding protein, or we can use the different kind of tags to purify the protein with a short period of time. Also reduce the cost of the production, so that we can easily commercialize this product, so that these plant-based products can minimize the product cost, so that anyone can use it, like the Asian countries or third world countries who are the very poor, who cannot afford the drugs or vaccines. Those kind of things can, the plant system can be a alternative to produce with a very low cost and industrial scale, so that they can also benefit. So our research should be benefit to the humans. So that is the one, that only our research will be a very great. So otherwise we do great research, but that is not useful for the human beings, or there is nothing outcome. So though we need basic research, but still we need to excuse me. So still we need this kind of system to produce the proteins. Okay. Oh, next here, what kind of products we can make? So that's important. So we have lot of molecular products. We can get from vaccine to antibodies, therapeutic proteins, nutraceuticals. So these are the different proteins already expressed in the plant system. They already tested their activity in the different kind of assays. By using that assay, they used insulin, they used BSA, even boy serum albumin, complement antibody complements, immunoglobulin. There are a lot of kind of things they have been expressed and already tested Most of the proteins are very active. It can be used for the human beings or animal products or natural herbal products. Anything you can use it. So these are the brief of different kind of field, different kind of things we can be used as a plant. You can use to produce plant as a bioreactor to produce compensate this kind of things. So these are the things are right now. There are various companies that are producing Pfizer or Merck. The different kind of companies, different products on pipeline. So some are already completed clinical trial, one or two remaining things are going on. So I hope soon many products will come from the plant-based products when compared with other eukaryotic systems. So that when you come, the products coming from coming from the plant-based engineering, it can reduce the cost of the production. It also reduce the drug price, so that common people can easily buy and they can increase their human wealth. I mean, global wealth will be increased. So here also different kind of LTP from the capsid protein, different kind of antigens or clinical going on. So soon these may be some proteins are already coming out. So they are waiting for the approval to for the commercialization. Remaining some must redo the clinical trial and phase 1, phase 2, that kind of things. So first human trials, the product from the plant drug, which is approved by the FDA, FDA, Federal Drug Development. So from the USA, the USDA they approved the first treat the type 1 culture disease with a Protolex company from the Israel company. They use plant cell suspension culture to express the character cell suspension culture, though produce this protein. This is the first human drug approved by the FDA. So next is also the different kind of Newcastle virus for the animal vaccine. They also first animal vaccine which approved by the FDA. This is how the Ebola vaccine, as I said, the process. Ebola, when the Ebola virus spread across the African, those time they're using the same transient expression system to produce the antibody, or they also produce vaccine for the Ebola. So here just a small how the different kind of storage pattern extraction pattern, just showing the from the, even we can use the corn just on the field, we can show the transgenic field and harvest, and you can usually use it as a medical. So such a very beautiful system, that is a plant made pharmaceuticals or plant system which can be enabled with any barriers. It can no barriers. Your gene of interest is everything fine, it can be easily expressed and purified, and we can send to the target people who are need for the things with the less price. That's very important. This is how they do the clinical trial, different kind of things for the global improve the global health through plant biotechnology or plant molecular forming or plant made pharmaceuticals, anything you can call it, using the different kind of, you can use corn, you can pills, you can use drinks, just a soup like that. You can also use a tomato, just so you can eat bite, bite, 3 bite, you can eat anytime, you can eat anytime, you can improve your health, global health. So these are the things, application for the how what kind of edible vaccine can be used for the different country, because there are so many things, so many viruses. So far, still we need a very cheapest to vaccine. So this kind of things are developing, and it's most of cholera retrovirus, Norwalk virus. These kind of things majorly have affected this. Okay, so dengue fever virus, we have a lot of virus. Even we have this Japanese encephalitis mostly in India. So that's a very worst kind of virus which can cause the lot of babies, like born babies 1 year or less than 1 year. There are a lot of people died in the Uttar Pradesh or Madhya Pradesh. If you see those places, Japanese encephalitis are very worst kind. We can easily produce the vaccine, or we can expose any antigen. It can be produced protect the humans values. So we can easily using the plant system we can do. Also another one company is the Protolex. That's what I said, it's Israel company which is first treatment drug from the character suspension culture. And next is a deconstruction for the icon. So they are also using the virus-based vector. They're using the producing antibodies or some kind of a hair non-human live vaccine. They are producing those kind of things. The hair the what is called human epidermal growth factor 2. They are also using the castle veterinary, so that university they are also producing for the, so this is also antigen presentation, so it can produce the epitope on the surface of the virus particle. Very important. So very easy. They are producing. They are going to soon. They are going to commercialize this kind of vaccine. So this is a different kind of vaccine. How they used for the tumor, any kind of things you can. Because see, nanoparticle, right? Plant virus is a nanoparticle. You can deliver the drug, or you can target any specific site. If there is some tumor, you can coat the tumor kind of compound or drug. You can directly target the blood through bloodstream, or you can target anyway, either way. So you can use this plant virus nanoparticle as a drug delivery system, so vehicles. So that's also can be used. So what is now we talk about different kind of things. Now we come to the commercial based. Okay, someone want to be entrepreneur, or some people, biotechnology students, he want to entrepreneur, he want to collaborate with the biotech like Bharda University or Medra University or Bharda University with the plant biotechnology scientists or researchers or professors. He has to know about the what kind of opportunity. So because most of the biotechnology students, we are seeking for the jobs. Instead of seeking for the job, we can start the biotech company. So how we can come biotech, because we cannot put a lot of money on the R&D. So we can collaborate with some research station like Biotechnology Institute or Botany Research Institute in the Bharda University or any other university. So you have to know the values of the, so far 2001 to 2020, it's about 158 billion. So the big scope for the commercial products. So you can, if you want to be an entrepreneur in future, because you study biotechnology but you don't know anything, so you can have this very good data. You can also find out in the NCBI. You have a lot of reviews on that. So these are the somewhat biotechnology company which is focusing on the various kind of biopharmaceutical proteins, vaccines, everything. Here, this bio app, this is company is owned by my professor in Dr. Huan Huang. We categories we have. First is animal vaccine, because animal vaccine is very easy to get approval. So we focusing on the animal vaccine. Second is the biopharmaceutical, like fibroblast growth factors and stem cell factors, this kind of growth factor biomaterials, which can be used for the stem cell research. So for stem cell research, they're spending a lot of money on the growth hormones. So we also produce a growth hormones. Apart from growth hormones, another kind of set of people, they produce the industrial enzymes. So industrial enzyme can be used for the different kind of purpose. You know that, as I said, you can use for the bio waste, organic waste management. There is a industrial scale like a paper industry, any industry which is required for the enzymes, xylene, cellulase, cellulose, a lot of enzymes available. So you can use any kind of thing. So we have different kind of application in our company. This is yeah, of course, in future, this plant-based expression system or plant-based bioreactor going to be future for the production of any biologically important pharmaceuticals or protein or vaccine or enzyme. So so far the evolution of the drug industry, every 20 years or 15 years it's changing. Now in future, plant will be the best and the future, and definitely it can be most of the people can get a lot of job opportunities. At the same time, also people can be a entrepreneur to bring their products. What in every country have their own requirements. You can also think about what are the requirements in India. You can just get touch with some scientists in the universities, collaborate with them, and you can make your own product, and you can have the small place enough to produce any kind of things. For the initially try with some kind of animal products, so that it will be very easy to get the approval from the government. You can have the very great future. Thank you. Thank you very much. If you have any question,
Yes,
please
yes,
kindly
yes.
ask me.
Thank you, Muthamel sir. Thank you.
Dr. Muthamel, can you hear me? Guru?
Yeah.
Yes.
Yeah.
I request Vasant, you can go ahead with the questions. Sir, can you hear me, sir?
Yeah, of course, I hear you.
Sir, thank you for your fruitful lecture, sir.
Thank you very much.
So I think so. This is the good time to share our knowledge.
Okay.
So there is some of a few questions from Pradap Bio,
Okay.
sir.
Yeah.
The nuclear transformation have some difficulties, but I don't know what is the reason behind that.
What kind of difficulties there? I mean, recently we have not that difficulty in transferring the plants, because when I was a student, that time it's very difficult to transformations. Even you don't know how whether the gene of interest is transformed or not. Now we have GFE. You can put the GFE along with your gene of interest. You can easily just put your light on UV. Definitely, if you are protein express on that, you can see the color, green color. So very easy right now. But I don't know what perspective he asking about the why difficulties. So that's I don't understand, because we don't see it, because every 3 months we in our lab we make 10 to 15 random transgenic plants, different kind of genes. So we don't feel any difficulties. If he has some difficulties, let us know. Maybe we can advise him what kind of things we can do. Otherwise, we need to know what kind of protocol he used, how he used to develop the transgenic plant.
Thank you, sir. Is there is another question also from the Pradap Bio? How to rectify vector contamination?
How to, but be a very professional, that's it. How can the vector will be contaminated? Be professional, pay attention where you are putting your things, that's it. If a contamination that's a man-made mistake, right? Vector cannot go each other and interact with each other contaminated. So we make contamination, or by mistakenly or unknowingly we do that things. So pay attention, that will be okay. Or
thank you,
maybe
sir.
does he asking about cross contamination or just vector contamination? Yes,
sir,
maybe
it's just vector contamination, sir.
Okay, yeah, vector contamination. Okay, cross contamination mean, of course, that's a different kind of questions. Okay, that's fine.
Thank you, sir. Is the question from Dr. Archana Meshram? Are plant-derived biological drugs as safe as other biological drugs?
Yeah, plant-based is one of the best, because we choose plant because of the safety. Because we eat plants, right? It will not cause any problem. When you eat too much of plant, that maybe it has some kind of. But we choose most of the industry. If you go to German, USA, they put lot of money on the plant-based pharmaceuticals or plant molecular farming. Because why they feel it? Because it can be very safe compared with any other eukaryotic system or a prokaryotic system. So, for instance, the mammalian cell culture, you can lot of prions available. There are virus, the toxins are co-purified with your protein of interest. Whereas in plants, it doesn't. So far, there is no report. So far, there is no any cases or like it has any kind of side effects or it has toxins. So plant we mostly using first is very cost is very cheap. You can produce any product very cheap. Second is a very safety is a most important factor. So very safe. You can use any kind of protein, anything. Yeah, it's a very safe.
Thank you, sir. Is there another question from Pradap Bio? In computational biology, some changes is there like genome analysis, annotating, pairwise and multiple alignments. However, how to cross over this much of research orientation problems?
Oh, I mean, what kind of research? I mean, I don't understand his question actually. What problem he has the computational?
Sir, I can repeat the question again, sir.
Question I understand, but his mind I don't understand his what he really want from me. That's what I don't understand. Because it's any competition like he want to check the okay, maybe repeat one more time questions.
Sir, I can repeat one more time, sir.
Yeah.
Yes, sir. In computational biology, some changes is there like genome analysis, annotating, pairwise and multiple alignments. So, however, how to cross over this much of research orientation problems?
Research oriented problems.
So
what he is asking us about system biology thing like that. Like we are doing genome analysis and annotation. like that.
Okay.
Yeah,
But
we'll be aligning multiple sequence. No, sometimes
So-
maybe when we are trying to express a protein. So his question, I think it should be like that.
yeah, so the point is when you decide a protein, for instance, you need to stem cell factor. You want to express the stem cell factor. Okay, you have to check first. Put in the NCBI, retrieve the sequence. You can have the different kind of organisms, have the different kind of sequences. Maybe it has a one mutation or some kind of sequence will be vary. So these are very simple processes, a bit computational alignment. But I don't understand his question because very simple I mean, it's a very simple process. When you have decided this is what I want to do, then the process is very simple. You can just take the sequence, you can go to the UniProt, and you can have most of the proteins are stored in the UniProt. So you can easily retrieve the protein, it available all the details. So it's not a much complicated. These days, there is no complicated. Maybe 5 years, 10 years, of course, little complicated. We don't know what kind of genome exactly we are taking right protein or right genome sequencing or protein sequencing. We don't know. But right now, most of the proteins so far published in the research journals, mostly we available in the UniProt. You go to UniProt, type your protein of interest. They will come a lot of different kind of forms, cytosolic or nuclear protein, whatever proteins. Then you can just easily you can get it. So very simple. It's not a big deal. Yeah.
Thank you, sir.
Mm.
Another question, sir.
Mm.
From Gayatri, apart from the potato, is there any available vaccine in the market?
No, right now we are on research Gorilla because I showed us there are 4 or 5 proteins. So that is the only protein available right now. Remaining are on process. Definitely in future, most of the protein will come. Right now, all on research or phase clinical trial or this kind of things. So not available. Commercially not available. Yeah, you can go to the next question. Okay.
Vasant, is there any other questions? Vasant, you can hear me?
Yes, no question.
Hello, sir.
Okay.
Hello.
Yes.
Sir, tell me about some of procedures for the hormone extraction from the plants.
Hormone extraction?
Methods, sir.
Oh, it's for instance, what we do, we use the put our gene of interest in that. We put some tag like a CBM3. CBM3 is a cellular binding domain. We co-expressed with the plants. Okay. In between, we also put some kind of cleavage. So we call it smoke cleavage. So we also put that protein. So we just express the plant and extract the total extract. From that, we use the cellulose beads to bind with specifically CBM3. After purifying after go through the process, because that's a big protocol. If you want, I can share the protocol. So go through the protocol because CBM3 specifically binds with the cellulose beads. So after pelleting the beads, so my protein also co-purify with the CBM3 because we fusion with our protein. Once it purified, then we express some enzyme. We cut with that enzyme that will cleave my protein and the CBM3 fusion protein. So separated. So we directly load into the gel filtration. So we separate our protein and that protein. After concentration, we quantify the protein. It's a very simple process. So it depends on when you design the things you want to express one protein, you design what kind of things, how you want to be a protein. You want to fusion protein or you want to express only one gene of interest. It depends on what you want. It's all about based on our need. So according to our need, we need to design the expression cassettes. Is it okay for you? You understand, right?
Yes, sir.
Yeah, it depends on you. If you want a design, you can design. If you want any advice from us, of course, we can guide you to how to design the expression cassette what kind of tags are you can use for the purification, how can you simplify the purification pattern. These are theoretically very easy, but when you do the purification process, it takes a lot of time, because that's what we are doing now. Compared to 5 years, now we improved a lot the purification system. Maybe another 5 years, 10 years, the technology will be very, very improved. So future will be very good. We will have the very good technology.
Sir?
Yes.
Yes.
Any
We'll
other
conclude
question?
the session, sir.
Guru?
Yes, sir. We will conclude the session.
Yes.
Yes. Thank you so much, Muthamil Selvan, sir. Muthamil
Thank you,
Selvan, sir.
Dr.
Very interesting, very
Okay.
informative session. You explained
you.
very well. Most of the participants are share their post in the comment box. They
Okay.
commented a very informative, excellent illustration and explanation. Very good, sir. Very clear. And some of them are asking to request your PPT.
Oh, sure.
sharing.
I already give it to Dr. Arun, so maybe he can share.
Yeah, sir. Yeah, sir.
I will share.
Yeah, sir. Yeah, sir. Also, very, very good and informative session, sir. I hope the participants are enjoying and gain more knowledge. And also, he explained very well about the advantage of plant expression system and also different types of expression system in plants. It's all are new things and
Yes.
molecular farming. Very interesting, sir. I hope all are gain more information from you. Okay.
Thank you.
So on
Thank you
behalf
very
of the Department of Botany and Biotechnology, I would like to thank you so much for take part in this webinar, sir. Thank you, Muthamil Selvan, sir. And I request to Dr. Jayabalan, sir. Please share your ideas. Yes, sir. Thank you.
Actually, I am proud about. I am the student of Jayabalan, sir. When I get the PhD, I got the recommendation letter from Dr. Jayabalan, sir. So I'm thank you.
Yes, I'm
thank
very,
you, sir.
very proud. Sir, your talk is very nice. Very up to
Thank you.
mark, you know. So
Thank you, Professor.
actually, when I was giving seminar at the time in the Netherlands, Wageningen University only produced vaccine in the honey based flower.
Okay,
but
Professor.
you are talking now. It's a very advanced stage of protein producing proteins.
Yes, Professor.
From the plant. Very marvelous. Really, I don't know where you are studying. You have your very good. You know,
Okay,
the expression of your talk.
Okay, Professor.
Really, I am enjoying your thing.
Thank you, Professor.
Really
Thank you very much.
happy.
Okay. Thank you.
Thank you so much, sir. Gurusaravanan. Thank you so much, sir. Guru? Dr. Guru? Tamil, we can hear.
Thank you so much, sir.
Guru,
sir, thank you so much.
Yeah. If you can give me some few minutes, am I clearly audible?
Yeah. Very clear.
First of all, I should be really sorry because I was not able to introduce the speaker to you properly. So if you give me 2 minutes, just I will brief this. I will try to introduce him to the speakers within 2 minutes.
At the end of session, we will introduce again.
because it will not be perfect, or I will not feel complete because that was not it was a technical error. But still, my mind is not getting it settled because this kind of a researcher like Muthamil, like every person has to know. So that's why, anyway, that I feel there is no order. Like we go in the reverse also, but the message has to be conveyed. No, the right time at the right message should reach the right people. No, so if I am very clear, then Dr. Muthamil did his PhD in National Chung Hasin University in Taiwan. And he initially, for his PhD, he was working in developing some novel transgenic cell suspension culture for producing virus-like particles in using plant culture. So for there itself, his research interest started to use plant as a factory to produce this kind of human proteins. And after that, he joined as a postdoctoral fellow in Taiwan. It's a reputed institute called as Academia Sinica, where he was working in the herbal medicine department. And there he was working for around 3 years. And once after that, he moved to South Korea. So there only he is currently now working in South Korea. Now he is like involved in developing plant-based vaccines for human papilloma virus. And recently he's also involved in development of plant-derived biomaterials for stem cell culture medium. You see, normally for culturing the animal cells, we have a lot of culture medias, right? But they are now designing a new culture media to like grow this kind of plant, like reformed or transformed cells. So really, like I am very much proud, Dr. Muthamil. So to be a brother, your friend. Okay, so you have done a very good presentation, and almost I can see that almost all participants were asking for your slides, which they are saying it was a very clear presentation. So if you don't have any problem, can I share your slides to the participants?
Sure, no problem, no problem. That's
so with your permission.
No need permission. Just a share. No.
Okay. Thank you so much for your gesture, Dr. Muthamil. So thank you so much, Guru. If it's okay, then we can conclude the session. Then okay, you can also say about the next day like session.
Yeah. Actually, tomorrow we have 2 resource persons. They are going to talk about bioelectrochemical system in aid of bioenergy and biofuels production from waste. The topic will be given by Dr. Chandrasekhar. And another one more topic in afternoon session, Dr. S. Vinod will going to deliver the lecture of technologies for graft genetic improvement. The session may be started at 3 p.m. Evening session. Then we have an conclude part that is validatory function at 4 p.m. That is tomorrow schedule, sir.
Okay, thank you so much, participants. Thank you, Dr. Muthamil.
Thank you,
participants. Thank you, participants, for spending your valuable time. Thank you so much, Muthamil Selvan, sir.
Thank you.
For spending your time for us. Thank you.
No, it's my great pleasure.
Thank you. Jayvan, sir, thank you so much, sir. Always be part in the session, all the session.
Thank you so much,
very
sir.
happy,
Jayvan,
sir. Very happy.
Thank you so much, sir. Jayvan, sir. Thank you so much.
Really happy. You know, my students, the students are well.
Really true, yes, sir. Because we all studied from you.
Yes.
When we are students, we listen your classes. So when we teach and you listening, that's for us a very proud moment, actually.
You are very good teaching capacity. Really, I wonder. Yes.
Thank you.
Yes.
Okay, Dr. Tamal. Yeah, nice.
Okay, now. Bye.
Thank you, sir. Thank you, Thank you. Sir, thank you, sir. Okay. Yes,